Hello everyone. Today we are going to present on topic prediction of cancer cells using digital signal processing. This is the list of contents that we'll go through. Moving on to introduction. So what is genomic signal processing? GSP, as it is called, is the engineering discipline that studies the processing of genomic signals, which are measured events originating from DNA sequence. The analysis, processing and use of genomic signals for gaining biological knowledge constitute the domain of GSP. Over the past decade, significant discoveries have been made that provide a better understanding of genetic basis of cancer disease. It has been understood that DNA plays an important role in the study of cancer disease. Now, most cancers come from random mutations that develop in body cells during one's lifetime, either as a mistake when cells are going through cell division or in response to injuries from environmental agents such as exposure to radiation or chemicals. In extraction of DNA sequence using silicon CMOS technology, silicon FET arrays are used. Now moving on to motivation. So in the present era of technology, medical field has become one of the favorite topics for researchers, and cancer is among one of them. It is a topic of concern because actual treatment of this disease is not found till date. Patients having this disease can only be saved if and only if it is found in early stage, that is in stage 1 or in stage 2. To provide the appropriate treatment to the patients, there is a need for accurate automatic prediction system for tumor and cancer, as previous treatments are manual and clinical based. Analysis of genomic sequence using DSP approaches such as filters, transformers and models has been increasingly used to recognize the hidden features and information of this sequence, which cannot be achieved by the traditional biological methods. So this is a literature survey. So starting with the first paper that we went through is the prediction and identification of cancerous cell using genomic signal processing. The advantages of this paper were they proposed a new method using modified wavelet transform for better prediction and identification of anomalies in cancer cells, and the disadvantage being cannot detect cancer irrespective of other abnormalities. The second paper was different genomic signal and processing methods for eukaryotic gene prediction, a systematic review. The advantages being interpret the results of data analysis using the developed tools in a biologically meaningful behavior, and the disadvantage of this paper was that it does not provide new technique to find cancer in its early stages. The next paper was machine learning application in cancer detection and prediction. The advantage of this paper was discusses the overall performance for each proposed scheme with their pros and cons, and the disadvantage was does not provide a method which combines various methods for better cancer prediction.
Okay, so now coming to research gap. Despite many advances derived from important innovations in technology during the last decades, in the field of cancer medicine, successes are still overshadowed by the tremendous morbidity and mortality incurred by these devastating diseases. So now coming to problem statement. Cancer is caused by abnormality in the genetic materials of the transformed cell. Cancer promoting genetic abnormalities may randomly occur through errors in DNA replication or inherited and thus present in all cells from birth. The cancer is usually affected by complex interaction between carcinogens and the host of genome.
So moving on to the block diagram. So this is a block diagram of DNA. So basically, what is DNA? DNA is a double helix structure with adenine, guanine, cytosine and thymine and hydrogen bonds between them. DNA is composed of sugar and phosphate with their base pairs with hydrogen bonds between them. They may be double bond or triple bond. Between guanine and cytosine, a triple bond is present, and adenine and thymine consist of a double bond. A sugar molecule with phosphate and any one part of base makes up a nucleotide. This is a molecular structure of adenine, cytosine, thymine and guanine. So moving on to the design methodology. A key concept in DSP is the possibility of representing the signals in the frequency domain, making the use of discrete Fourier transform. This representation leads to some of the important signal properties which are associated to the frequency spectrum that are not revealed in this time domain. In case of these genomic sequences, the nucleotide bases are represented mathematically by character strings of size 4 alphabet consisting of the letter A, T, G, C, which denote adenine, thymine, guanine and cytosine respectively. The possibility of finding wide applications of DSP techniques to the analysis of genomic sequences occur only when these are appropriately converted into numerical sequences. Several mapping techniques are used by researchers for the applications. A new mapping technique of conversion based on weak and strongly hydrogen bonding between the nucleotides bases has been used in this paper. Here, instead of taking the 4 independent indicator sequences for each nucleotide, one binary sequence is generated based on weak and strong hydrogen bonding. For the conversion, we have taken A, which is adenine, and thymine to be binary 0, and cytosine and guanine as binary 1. So, for a DNA string which is defined as x of n, consisting of alphabet A, T, C, and G, of length n, let us define a single binary indicator of sequence xs of n. X of n consists of these elements, so they can be represented in binary form as A can be represented as 0, T can be represented as 0, G as 1 and C as 1. The DFT, the discrete Fourier transform of the corresponding binary sequence is, using the formula of x of s n into e to the power minus j into 2 to the power nk into pi divided by n, where k ranges from 0 to n minus 1 and n ranges from 0 to capital N minus 1. So finding the power spectral content of the sequence is given by ps of k equal to summation of x of a k whole square. The plot of psk of the coding region is investigated as indicator of cancer or non-cancer cell. So the algorithm of the code. For a coding sequence of length n, we need to find the power spectral density. First step, we need to scan the sequence and map the 4-letter alphabet into one binary sequence based on weak or strong hydrogen bonding between the nucleotides. Second, obtain the discrete Fourier transform of the binary sequence using equation 1, which is the equation of the Fourier transform. Third, estimate the power spectral content for the sequence obtained in equation 2. Compare the PSD for the cancer cell and the non-cancer cell. The algorithm is tested for various Homo sapiens. Cancer and non-cancer data bases are available in GeneBank, giving the satisfactory result.
Now I will be explaining the program which has been given in the previous algorithm. We are using getGenBank function, which actually takes the information from a website called as NCBI, which we have to give the accession number of each DNA sequence which we are trying to find, and the sequence only, and the partial sequence is given. The sequence only takes the sequence of the DNA from the accession number, and the partial sequence which we have stated in this as 4453 to 5157 takes the sequence from that number, that range. Then we convert the given sequence into its conjugate binary sequence, where A and T is given as binary 0 and G and C is given as binary 1 for the respective hydrogen bonding. Then we want to convert the given sequence and transform it into its FFT. Then we have to find the power spectrum density, which gives a clear picture about the DNA sequence and the various peaks and troughs in the given magnitude and phase spectrum. Now we are taking frequency where we want the range of the given x-axis and y-axis to have the same vector length. Then the subplot of both cancer as well as non-cancer characteristics are shown. In the cancer characteristics, we would like to see the x-axis as normalized frequency and the y-axis as absolute square. Then the axis length for the x-axis is taken from 0 to 1 and the y-axis is taken from 0 to 0.1. Then the respective characteristics of cancer cells and non-cancer cell characteristics are shown.
This is the obtained output characteristics. This is a cancer signal characteristics with the amplitude square and the normalized frequency, and this is for a non-cancer signal characteristics. As stated before, the cancer cells are mutated cells which absorb energy, thereby in sudden frequencies there is a sudden spike in the amplitude, whereas in non-cancer cells there are less or very few mutated cells, thereby there are lesser number of peaks, and the given characteristics as follows. Now moving on to the finding delta X improvement. DFT power spectrum plot can be used in an effective and a simple method for cancer prediction if DNA nucleoid sequences are available. In most of the cases of cancer cells, spikes are detected in power spectrum plot which is absent in normal cells. By using filters, we can have a more clear distinction between cancer cells and normal cells. These are the list of groups working globally in the selected area.
So now moving on to conclusion. DSP nowadays plays an important role in DNA sequence analysis, cancer diagnosis and gene expression analysis, etc. Researchers are using DFT power spectrum plot to predict protein coding regions of DNA sequence. Here we surveyed DFT power spectrum as a method to predict cancer diseases for various databases available in GeneBank. The result shows this method can be used as an easy tool to predict cancer diseases. The filtered power spectrum plots yield high accuracy. The bar plot shows prominent result for predicted of cancer. Further efforts will be made to improve the accuracy of prediction by using other types of digital filters. And the next step in our research would be to generalize the analysis for a number of other oncogene database. So now here are some references which we have referred, and this is the link to the individual subjects. Thank you.
So this PPT was presented by Kshitij Parashar,
Prahlad Inani,
Murli Ganesh,
and Madhul Mayank.